Hello, my name is Steve Jackson. I'm the Associate Director of Product Applications here at Thermo Fisher Scientific. And in this talk, I'm going to talk to you a little bit about how you can analyze immuno-oncology-related gene expression patterns using tumor research samples using RFID-tagged TaqMan panels. And this exploits our new flagship Quant Studio 6.7 Pro instrument that has a lot of smart features, which I'll briefly touch on as a result of this talk. So one of the main problems that we're facing when dealing with cancer is that a tumor sample can be sensitive to growth deactivation using a number of immune system checkpoints. There are several different of these checkpoint pathways that exist. There's the CTLA4, there's PD-L1, there's a lot of different, well, I won't say a lot, but there's several different checkpoint pathways. But many times you are unsure which of those pathways are going to work to inhibit cell growth. So how can you tell which one is effective? One of the ways you can analyze this is by looking at the expression of subsets of genes in the tumor sample. And by looking at the subsets of these genes, you might be able to say which of these pathways might be effective in inhibiting growth of these tumor cells. Thus, there's a need to easily analyze gene expression levels in immuno-oncology research pathways. So over the course of this talk, I'll basically make use of an experiment that we did where we obtained 10 colon tumor and normal adjacent FFPE samples. We analyzed expression levels of these genes by looking at two gene expression signatures that were defined in publications that came out recently. These expression signatures are associated with immuno-oncological response. And then we correlated differences in expression signatures of these two groups of genes in 3 different comparisons. We looked at tumor versus normal. We looked at PD-L1 positive staining versus negative staining. And we looked at microsatellite stable versus microsatellite unstable groups of these colon tumor samples. So as I mentioned, we recently came out with the Quant Studio 6.7 Pro real-time system. This is our flagship real-time PCR system. And we have introduced several new features that make this easier for you to use in the laboratory. One of them is that it has facial recognition and voice commands, so it can respond to your face or voice commands, freeing up your hands for doing other things. It introduces RFID plate readers, so that on an RFID tag, there are certain things that can be coded and automatically uploaded into the instrument. I'll have more to say about that in a second. It's a true standalone operation with no external computer needed. It facilitates tech support by having direct contact with tech support and readily downloadable files by tech support team to help the speedy resolution of technical problems. And there are other things you can read here, block ease of changing, cloud connectivity, instrument connect monitoring. All these things help make this instrument very easy to use and take your PCR experiments to the next level. Now, as I mentioned, the Quant Studio 6.7 Pro comes with RFID tagging. This is a radio frequency tag that is embedded into a plate where the RFID enables a smart workflow. The RFID can include the plate layout, all the assay information, and run method, all in this RFID tag. It can get immediately transferred to the EDS file after the run is completed, and when your run is started, it will be uploaded to the instrument. So you just have to basically put it in and turn it on and go. The TaqMan assays in these RFID tag plates are pre-spotted on the plate, and so it's very convenient. Then you just add your sample and master mix, and you're off to the races. And then finally, the data analysis is very easy because you don't have to download TaqMan files and upload a plate layout. Once the experiment is complete, the data is immediately analyzable because the EDS file contains all the important information about the location of the assays and the run that was done. So it's a much simplified workflow as shown here on the left-hand side of this slide. So what we did was that we used these two publications to order RFID-tagged plates that had these two signatures embedded in them. One of the signatures was based on an Ayers et al. Publication that came out in Journal of Clinical Investigation 2017. This is a T-cell inflamed gene expression signature that predicted response to PD-1 inhibitors. Again, you can go back to the original publication to find out what these genes actually were, but this is one subset of the genes that we put into the plate. Another publication came out of the group Shukla et al. This was published in Cell in 2018. And in this publication, they looked at a set of germline antigen genes that predict resistance to CTL4 responsiveness. So 8 different genes that were indicative of how well the CTLA4 was going to be able to work in those cells. We also had housekeeping genes 8 different housekeeping genes that we included in the reference plate, and so we ordered these RFID tagged plates that had both the PD-1 inhibitors and the CTLA inhibitors embedded in them. We then analyzed those 10 tumor and adjacent normal genes, colon tumor samples, and looked at the differences in expression patterns that we were able to see. So at first, in the T-cell inflamed gene expression set, when we looked at tumor versus normal, we saw that there's a significant difference in the T-cell inflamed gene signature in the tumor samples compared to the normal samples. That we found by doing some statistical analysis of these that the difference was statistically significant with a p-level of 0.02. Here you can see that the overall signature of the genes, not every gene, but the signature in general was upregulated by reflecting its lower CT score. These genes were upregulated in the tumor cells versus the normal cells. So no real surprise there. We'd expect there to be some of the tumor genes that would be responsive to PD-L1. If we made the same comparison looking at the CRMA genes, the CRMA is what the other subset of gene expression, other subset of genes we were looking at the gene expression of in that TaqMan plate. We found there was no statistically different expression of the CRMA genes when we just looked at tumor versus normal research samples. So here we did not see that there was a difference in the comparison of just looking at normal versus tumor. Now, when we wanted to look at the PD-L1 positive tumor cells and see if we could see differences in expression if we now grade them or differentiate them based on PD-L1 staining versus no staining. And so we sent the samples off to a third party to get staining. Here we can see an H&E hematoxylin eosin staining of a normal colon, anti-PD-L1 staining of an adjacent section, and no antibody of an adjacent section here. And this is from the tumor that was adjacent to the normal tissue. Here we see the H&E staining. There's PD-L1 staining throughout this particular sample, and again, that's specific to the antibody because no antibody didn't show any staining at all. So now if we look at the PD-L1 positive versus PD-L1 negative staining, we can see that the T-cell signature is significantly higher in the PD-L1 stained samples versus PD-L1 positive versus PD-L1 negative samples. But again, when we look at focusing on the CRMA subset of the genes, we don't see a difference in CRMA gene expression when we look at PD-L1, when we compare PD-L1 positive to PD-L1 negative stained samples. Again, not a terrible surprise since we expect that there'd be some T-cell infiltration causing the upregulation of the T-cell inflammation-specific genes. Now, the last thing I'm going to talk about is the microsatellite status of these samples. For this, we turned to our TrueMark microsatellite instability assay. This is an assay released last year that is a way to look at 13 different microsatellite markers. It makes use of as little as 2 nanograms of FFPE-derived DNA. We have facilitated the analysis of MSA stable and unstable with customized software. And one particular aspect of the software is that we don't require there to be a matched normal sample for the analysis. We make use of an extensive database of normal samples to come up with the normal samples that our software uses. Now, again, in this case here, we did have normal samples that we were able to compare to and to make the microsatellite calls, and we did that for these experiments I'll tell you about here. Overall, you can extract DNA and get an answer in as little as 4 hours with less than 1 hour hands-on time, and this runs on the Seq Studio and other 3,500 genetic analyzers. So it's a CE-based microsatellite instability assay. And so we use this assay then to differentiate microsatellite stable from microsatellite unstable tumor samples here. And when we look at the MSI, when we look at the T cell inflammation gene expression signature in microsatellite high versus microsatellite stable samples, we don't see a difference in expression of that particular signature. But when we look at the signature of the CRMA genes, we do see that there's a difference. In the microsatellite unstable samples, we see that there's a lower expression of the CRMA genes, whereas in the microsatellite stable tumors, that there is an upregulation of this set of CRMA genes. This suggests that in the tumor cells that are microsatellite stable, they might be resistant to blockage using CTLA antagonists, whereas the microsatellite high samples might be affected by CTLA blockage. So this might be a way then to distinguish between the way that these cells might respond to a immuno-oncology blocker. This talk, we talked about ways you can use a TaqMan gene expression panel to differentiate the way that tumor samples might be able to respond to an immuno-oncology treatment. In these experiments, we made use of some of the smart features of the Quant Studio 6.7 Pro and our RFID-tagged plates that really helped facilitate the speed at which this work was done, because we didn't have to mess with uploading TaqMan assay identifications and mapping them to a plate, et cetera. That was all done on the RFID-tagged. These RFID-tagged plates can be custom ordered from us, and they'll be pre-spotted with any TaqMan assay. I showed you that gene expression signature discovery and analysis can be an important part of the immuno-oncology research program. And panels that contain TaqMan assays for sets of genes can define a signature and can be used for these immuno-oncological research results. I want to emphasize these panels and everything here is for research use only and not for diagnostic use. Finally, I want to acknowledge Harita Veerashlingam, a member of my team who did all of the work here, and Kami Varma, on whose watch this work was performed. And thank you very much for your time.